Good morning, everybody. My name is Sergey Nikolenko, and I will be speaking to you about SPAdes, which is a new genome assembler that has been developed in our lab over the last year and a half, probably. And be warned that this was a last-minute decision to put me in here, because Glenn had to cancel at the very last moment. So if anything goes wrong, I'm sorry. Right. So SPAdes is an assembler which is which was first specific. Yes. Sorry.
Could you speak up a little bit? Yeah, the same.
Okay. I'll try, but I have a microphone. Does it do anything? No. Okay. So SPAdes was initially conceived as an assembler. As an assembler designed to handle single-cell sequencing projects. And single-cell sequencing is a way to get genetic information that we previously had not been able to get at all. It turns out that many bacteria, which have been called so-called dark matter of life, cannot be sequenced by standard techniques like Illumina because, well, most often because they simply cannot be cultivated. They cannot be separated from their biotas, and they cannot be cloned 1 million times individually so that we can get a lot of pure genetic material from this specific cell. And before the advent of single-cell sequencing, all we could do was to look at this sample and try to apply metagenomic analysis. And metagenomic analysis basically doesn't work too well. It does not produce finished genomes, and probably there is not enough information for it to produce finished genomes. So that is why single-cell sequencing has become an important tool over the last 2 or 3 years. And important examples of single-cell projects include, well, they began from more or less the modern state of single-cell sequencing began from marine cell sequencing, from deep ocean samples that are in essence metagenomic. And again, we can single out a cell from this sample and then try to amplify it by this single-cell technique, which I will get to a bit later. But in the last 2 years, there have been a few projects that dealt with specifically human microbiome and bacteria from the human microbiome, and even with such applications as sequencing tumor cells. So maybe, I don't know, but maybe single-cell sequencing is our way of understanding cancer and stuff like this. So I'm not a biologist by any means, so I will only say maybe 1.5 words on how single-cell sequencing works, and I will mostly concentrate on what are the computational challenges that it presents, and why is it more complicated, why is it hard to assemble genomes from single-cell data. Okay, so just as a quick reminder, what is genome sequencing, what is genome assembly? Usually, I mean, the DNA is the very long string which we cannot read directly. So what we do is we perform sequencing, which means that we break this DNA into a lot of small pieces, and then in software we try to piece these pieces back together into a single string. And this is called genome assembly. And for the popular sequencing technologies, the reads are usually from 35 to 400 base pairs long. And most often, modern technologies produce the so-called, in particular, the Illumina technology produces so-called paired end reads, or read pairs, which means that basically you have a longer piece of the genome, a longer sequence, but you cannot read it entirely. You can only read 2 small fragments from each end. So what in the end, what you know are 2 short reads and you have some idea about what is the distance between them. And this will be an important point later in the talk and in SPAdes. Right, so what single-cell sequencing is based on is the so-called multiple displacement amplification technology, the MDA technology, which basically it can basically it's called a Phi-29 polymerase, and it sits on the genome and it separates it into two strands, and then it sits on each of the strands, and the process is repeated, so you get a kind of an exponential blow up in the copying of the genome. So this looks like a perfectly valid way to get lots of genetic material, and then you can do it by regular Illumina sequencing or something. You can apply regular sequencing to get the reads. But this MDA technology has a few peculiarities that make our life of the people who try to assemble these reads much harder, right? So first of all, and the most important problem of them all, is highly non-uniform coverage. Coverage means that, well, coverage is the number of times each base in the genome gets copied in the reads. And in regular sequencing, in conventional multi-cell sequencing, well, you basically get uniform coverage along the entire genome. So there are variations, but they're very, 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 very small. In single-cell sequencing, you get highly non-uniform coverage, and you get, and this is a logarithmic graph actually. So you get entire regions which are not covered at all. Well, basically you cannot do anything there, so it's not a big problem. But the big problem is that there are that some regions are covered in the thousands, and some regions are covered in the tens or dozens. So what this basically means for the assembler is that most conventional assemblers simply use a coverage threshold to single out reads which are thought to be erroneous or which are thought to be chimeric or which are not good for the assembly. But if you, and for regular sequencing projects, it works perfectly. If you, in this graph, you could cut off and coverage 400 and not lose, mostly not lose anything. But for single cell sequencing, if you cut off at any specific coverage, basically, you get, you lose a lot of the genome material. And we will see that in the results.
What's on the axis there?
Now the y-axis is the percentage of positions that have coverage specified on the x-axis. So usually you have most positions covered around, well, in this case around 600. It's not a hard number. But in single cell, you get a lot of positions that are covered, that are very covered only a few times, and then you get a long tail. So you get some positions that are covered a lot of times. Right. So this was the first problem. The second problem deals with the insert size. And the insert size, if you remember about the paired end reads, the insert size is the length of this long segment from which we have read only two pieces on the left and on the right. So again, in regular multi-cell sequencing projects, insert size is not exactly what it is promised to be, but it is close, and it is a very, it is a very neat normal distribution, and you can, well, you cannot safely assume that it is exactly what you expect, but in most cases you can. In single cell sequencing, again, this problem gets a bit more complicated. So what you have is, well, at least in our data sets, what you have is kind of a bimodal distribution. And of course, there is a peak where it is supposed to be, but the variance is much larger, and the insert size variations are much more complicated. Finally, there are simply more errors. There are more errors, and not only there are more like random mutations not only there are more random mutations in the data, but also there are lots of so-called chimeric connections. The chimeric connection is when a read or a read pair joins up to far-off parts of the graph, and this greatly complicates the analysis. Right, so here again, to recapitulate, here are the 4 main problems of single-cell assembly. And historically, well, it's funny to say historically about 2011, but historically the first single-cell assembler was called E plus VSC, which stands for Euler plus Velvet SC, and reads like Euler plus Velvet minus single cell. And it was successful. It captured 25% more genes on single cell data sets than regular assemblers. But of course, our goal was to improve upon it. And in this talk, I will try to present some of the ideas that go into making an assembler that can beat E plus VSC and other single cell competitors. All right, so here is the general structure of how an assembler usually works. So first, you get the original reads. You try to correct errors, whatever you can. And then the corrected reads, you construct the De Bruijn graph from the corrected reads. And I will remind what a De Bruijn graph is. But I'm afraid that if you don't already know, maybe you will be lost there. So then on this very important step, you try to simplify the De Bruijn graph. So basically, you try to fix the errors that error correction has missed. And in the single cell case, error correction will miss a lot of errors. So then you get a simplified graph. And then usually what you do is now you begin to actually utilize the read pair information. So now you begin to use this information that reads come in pairs, and that we know what reads should be close to which reads, and we try to resolve repeats, and we try to further simplify the structure of the De Bruijn graph with these repeat resolution steps. And then there is a post-processing step in which you can go back to the original reads, try to map them on the context, and fix the few remaining errors. All right. So I will go through this structure from top to bottom, but of course it will be a very high-level overview, and I'm not going to prove any theorems, or I will try to present the basic ideas that distinguish SPAdes from what has been done previously. So, all right. Well, first of all, let me introduce k-mers. K-mers are the main building block that all assemblers work upon. So if you have a read, what you do is you break a longer sequence into shorter subsequences that basically overlap as much as possible. And these k-mers, where k is the size of the subsequence, the k-mers, well, the basic idea of modern genome assembly is to take the k-mers and construct the De Bruijn graph of them, and then try to find an Eulerian path in the De Bruijn graph. But of course it doesn't work, because there are all sorts of errors in the reads. And so the first step is to try to correct the reads as they come. So the first step, actually, if we did not have this error correction step, we would not even be able to construct the De Bruijn graph. It would be too, well, we would be able to construct it, but we would not be able to work with it. It would be too large and too complicated. So the first step is to try and correct the reads. And in SPAdes, error correction also works via k-mers. So again, if you have the original read, and you know how to correct each and every k-mer in the read, then of course it is easy to correct the read, right? So you can reduce the problem of correcting the reads to the problem of correcting the k-mers. And in regular multi-cell error correction in tools like Quake, of course what you do is you count the k-mers, and you look at how many of them you have, and then you say that k-mers that occur seldom are that rare k-mers represent errors, contain errors, and you throw them away, and then you try to correct the reads from the solid k-mers that are highly covered. Of course, in single-cell dataset, this does not work at all. So there is no global threshold that you can apply. So what you can do, and what is the idea of hammer, and which was originally presented by Paul Medvedev, who is here in the audience, and later extended into Bayes hammer, which is now a part of SPAdes. The idea is to look at k-mers locally. So if you look at a set of similar k-mers, then of course you can, well, I mean, it's still not a given, but you can kind of assume that they come from the same part. And if your errors are rare here, then you can correct them. So the difference between hammer and Bayes hammer is that Bayes hammer attempts to cluster these sets of closely matching k-mers into maybe not just finding the consensus, but maybe cluster them into, say, 2 or 3 clusters. Right. I think I'll skip this one. But as a result, our tool corrects single-cell data sets much better than existing tools that are targeted to multi-cell assembly. Sorry. All right. So once once we have corrected the reads, now the main idea of read correction, of error correction, is to the main idea of error correction is to get rid of erroneous k-mers. The main idea is to reduce the set of k-mers. So now we have, and actually, well, that's what it does. I mean, in single cell, in one example of our single cell E. coli data set, we go from 200 million k-mers down to about 30 million, which is a much more manageable number. And when we have relatively few k-mers left, we can begin looking at them on a global scale, which means we can begin to construct the Bruin graphs and we can begin the actual assembly. So, a quick reminder on the Bruin graphs. Here is a very simple example which represents a circular genome, and it is perfectly covered by reads of length 3, and we break each read down into 2 k-mers, into 2 tumors, into 2 k-mers of length 2. And then in the construction of the De Bruin graph, what we do is we identify identical k-mers. We identify k-mers with the same strings. And even in this perfect, very simple example, which is a very short genome, which is perfectly covered and everything is here, and the coverage is very uniform, even in this very simple example, what you get is basically a mess. What you get is a rather complicated graph, and it is by all means not obvious how to get from this graph, which is what you get in the beginning, back to this circular genome, right? And that is the problem of genome assembly. And of course, if there is a single string that unites all k-mers, it would correspond to an Eulerian path and an Eulerian cycle in this graph. And that is true anyway. But the premise of this does not hold. Because there are erroneous reads, erroneous k-mers, which lead to spurious vertices and spurious edges that should not be in this path, on one hand. On the other hand, gaps in the coverage lead to make this graph disconnected, and you cannot really even hope to find a single path that unites the whole graph. But so in the presence of error, genome assembly, finding an Eulerian cycle is a simple problem, but in the presence of error, it is a very hard problem, and basically it is actually not very well defined problem, especially if you cannot make assumptions on coverage. But still, we have to do something. And here is a nice picture of a connected component from one of our data sets. So here is the connected component itself, and here it is amplified couple of times, so you can see that it does have a complicated structure. Right. All right. So the first step of assembly is to try and simplify this graph. Of course, you cannot read the sequence from here. So to simplify this graph, we have to fight the errors that remain in the reads. And errors now on this stage, in the construction of the De Bruijn graphs, errors represent specific local constructions that can be identified and can be, well, kind of corrected. And there are three kinds of very common errors in the De Bruijn graph, and every assembler has to somehow handle them. The first example is a tip, and if you have a single error close to the end of the read here, then if a k-mer does not fit in here, then what you get is a tip. You get a spurious path that comes into or goes out of the correct path. And in most assemblers, what you do is you look for tips that have small coverage and you cut them. It's called tip clipping. But in single cell assembly, you cannot do that. So we have a novel tip clipping algorithm, which is enhanced by gap closing. I will say a few words about this later. Okay, the second part is a bulge. And the bulge results when you have an error in the middle of a read. So you have a k-mer on the left and the k-mer on the right. And so the wrong path starts from the right path somewhere and then comes back into it. So again, what every assembler does is it projects a wrong path on the right path, on the correct path. But again, in single cell assembly, we cannot just remove P, maybe increment the coverage of Q by 1 and be done. We have to remember what was here because it may be useful for repeat resolution later on. So so SPAdes incorporates additional bookkeeping that preserves these mappings of the bulges. This is called bulge core removal, which stands for bulge correction and removal. And the third common error, which is not all that common in multi-cell assembly, but which is very common in the single cell case, is a chimeric connection. And the chimeric connection joins two unrelated parts of the graph. A chimeric read is a read that basically just accidentally has two parts from different parts of the genome put together. But in single cell assembly, this happens a lot. So we have to somehow handle these chimeric connections. And SPAdes, again, I will not go into details here, but SPAdes uses a novel algorithm for dealing with chimeric connections. And we have tried approaches based on MaxFlow algorithms. I'm not sure if they're actually working in the code right now, but there are many ideas here. And what is even more important is not how you solve every single local problem, but how you go from the local problem, but how you find these local problems in this whole global entangled mess. And to that end, SPAdes uses special scheduling algorithms, which are also a novelty, that attempt to find the correct order in which to handle these common situations. For example, in this case, of course, we immediately see that you first clip this tip, then you project this bulge, then you project this bulge, and then you're done. But in software, and when you're like locally in this vertex, it is not immediately obvious, right? To sum up this part, for single cell data sets, errors become harder to correct because you cannot rely on coverage. So we have developed several new algorithms that process errors based on graph structure and not only on coverage, although it still plays a role. And also a very important point is that SPAdes processes errors in a specific order that results from special scheduling policies. Okay, next idea. Let us again go back to this simple example of a circular genome, and let us vary the value of k a little bit. So first, let's cover this genome by reads, and let us consider that the Bruin graph for k equals 3 here. So for k equals 3, you get this entangled mess which you do not know what to do with. On the other hand, for k equals 4, you get, in this example, you get very simple connected components in the graph. You get a single straight path that unites everything, that unites half of your genome. But the problem here is that the graph has become disconnected. And because you have increased k, you have introduced gaps in the coverage with this k. Right? So there is a trade-off here. For small k, you have a highly connected but entangled graph. For large k, you get a disconnected but less entangled graph. So so what SPAdes does here is to use the so-called multi-size de Bruin graph. At least the idea of the multi-size de Bruin graph, which was introduced by IDBA assembler in 2010. The idea here is that you first construct. The idea is that you construct a graph for a large k, then you try to close the gaps based on the graph constructed from smaller k. And this is what SPAdes does also. Right. Let us move on to repeat resolution. And for repeat resolution, I first need to introduce to you what a repeat is. So, well, a repeat is a repeated sequence in the genome. But in the de Bruin graph, it results in an edge that should be somehow traversed several times. So the correct path, it is not Eulerian anymore, the correct path should go through this edge several times. Sorry. Like here and then there. And the problem here, the assembly problem here is how do you match the incoming edges with the outgoing edges? So suppose that you know everything except that you have a single path here and a single path there, but how do you know if you come from here, how do you know where to exit? And this is crucial for getting long context. So some repeats are easy to handle. For example, if you have a single read that goes from here and comes all the way to here, to the blue guy on the bottom here, then of course you know that one path comes from here and exits there, and by elimination the other path goes up. But there are many more complicated repeats, and in a more complicated repeat you do not have this kind of information, and you cannot just conclude this. You cannot just make this very simple conclusion. So, so what you do in this step is you recall that the reads actually came in pairs, and most repeat resolution strategies are in some way or the other based on using paired information from the reads and somehow matching this incoming and outgoing edges via this paired information. And the basic theory, again, there is a beautiful theoretical construction that greatly helps our understanding, but doesn't really help our algorithms, which is the so-called paired de Bruin graph. And a paired de Bruin graph is, again, a de Bruin graph. It is constructed in exactly the same way, but now we change the definition of a vertex. So if previously we had vertices corresponding to k-mers, now we have vertices corresponding to pairs of k-mers. So if you have a paired read, and if you know exactly the distance between this pair, the insert size in this pair, then of course you can match this k-mer with its counterpart, and you can say that, okay, we have a k-mer ATC and the k-mer GAT, and there is a distance. What is the distance? 1, 2, 3, 4 between them, right? And if this distance remains constant throughout all of your reads, then you can construct a paired de Bruin graph. So you can now you do the same construction. Again, you break up reads into k-mers and you identify identical k-mers, but now you identify only identical pairs of k-mers So you get a lot less entangled construction. And if you recall, there were a lot of individual k-mers that could be identified here. But as a pair on a certain distance, there is only one pair which matches perfectly. So the paired de Bruijn graph for this example looks like this, which is of course much simpler. And probably when we are looking at tumors, we cannot do any better than this. Right. Of course, in theory, we would construct the paired de Bruijn graph, and we would find a path there, and we would say, and basically, you can safely assume that if everything is perfect, then the paired de Bruijn graph incorporates all information that you have from the paired reads, and it is a very nice construction. But of course, in theory, there is no difference between theory and practice, but in practice there is. So, and again, this is a major problem for implementing paired de Bruijn graphs, and it is a major problem in many aspects of genome assembly. And the problem here is that the insert size is not constant. If we knew exactly what is the distance between paired reads in a pair, then we could do it directly. Even with all the errors and with all this stuff, we could simplify paired de Bruijn graphs, and we could make this work. But the problem here is that the insert size is not constant. And this is a sample empirical distribution of insert sizes between two relatively long edges in the graph. And so what SPAdes does here is SPAdes attempts to estimate this distance between this insert size by clustering this empirical distribution and adding the graph structure into the mix. So in this picture, what you see is basically there is a very long, the numbers here are sizes or length of these edges. So you have a very long edge coming in here, and you have a very long edge coming out there. So you're basically certain that you come in here once, and then you come out of there once. But the question is, the question of resolving this loop is whether you go straight there, and this 13 is just an error, or whether you should circle back and then go out. Okay? And if you look at the distribution of the paired end reads and paired end distances, how they align on these edges, then you see two clusters here. You see that there is one cluster that corresponds to paired information coming from here to here with distance 0 here, and another cluster of paired information that comes from here to here with distance corresponding to this loop and then this edge. Okay, I don't want to get into the numbers, but basically this histogram shows that you do have to go around this loop at least once. And if your insert size is long enough, then you can say that only once. If it is shorter, then you cannot really say if you have to do it twice. But basically, what you can conclude is that this 13 is not erroneous. You have to go around. All right. Next idea. It is a tight talk. The next idea deals with a process called gap closing. And gap closing in regular multi-cell assembly, what gap closing is that you construct your de Bruijn graph, you clip all the tips, you simplify it, and then what you are left of are several connected components of the de Bruijn graph. And it turns out that sometimes you can join these connected components. They are disconnected because there is no k-mer in the reads between them. So there is no path in the de Bruijn graph between them. But maybe there is an overlap which is just not big enough. So you can identify, first you can identify these overlaps. And then you can see if they're supported by paired information. If you have several paired end reads such that one half of the pair maps here and the other half maps here, you can be pretty sure that this was a single string all along, and you can join this to paths. And this is called gap closing. This is called gap closing with read pairs. Now, in SPAdes, in single-cell assembly, we use this idea also for identifying erroneous tips. So this, in regular multi-cell assembly, you can clip the tips based on coverage. In single-cell, we clip the tips based not only on coverage and graph structure, but also on whether we can close the corresponding gaps in the graph. And so the gap closer, our gap closer actually runs before tip clipping. And many of the tips turn out to be actual discontinuities in the graph. All right. So now that we have, now that I have explained, well, at least began to explain some of the complications and some of the ideas behind this, we can go back to this graph, to this connected component, and we can see that after all is said and done, and after all SPAdes algorithms are applied here, we are left with this. And this is actually a relatively old picture. I think it's from 3 months ago, so now maybe it's even simpler. Maybe Sergey can tell me. Basically it works. Now you can find long paths through this very simple connected components. Right. So there is one more idea that I wanted to at least mention before I conclude my talk. And this is a very recent development in the lab, and it was mostly developed by Kolya Vyache and recently published. And this is a slightly different way to present the paired de Bruijn graph and to present the paired information in the reads, which turns out to be a useful way to represent them, especially in the presence of errors in the distances between k-mers. So this is called a rectangle, as you probably see. And what we will try to do is we will try to construct, again, a kind of a de Bruijn graph, which is called a rectangle graph. It consists of this rectangle. So a rectangle is a pair of edges in the de Bruijn graph such that we have paired information between them. And we have an estimated distance in the de Bruijn graph between these edges. And now this blue edge, this blue diagonal here, it comes through these pairs of k-mers that are on distance d between these edges. Okay? So basically this blue diagonal in the rectangle is a path in the paired de Bruijn graph, basically, for this distance d. But now it is a bit more. But now we have a bit more information because we also have these candidate neighbors of this path, and in case we have a diagonal coming through here, and then we have a diagonal starting out from here and going through another rectangle, we can kind of assume that maybe there was a plus one error in the distance size, and maybe we can join them in the path here. So as a result, you get a so-called rectangle graph, in which you have all these rectangles. Rectangles correspond to condensed edges of the de Bruijn graph. You have all these rectangles, and you identify and you connect the blue dots that are at nearby places of matching edges of the rectangle. And then the problem is again to find the path through all these rectangles here. Okay. No, if the edges had the same length, it would be square. If all distances were the same, then all these blue dots would match perfectly. But the real situation is that you have a blue diagonal here, and it may match to a blue diagonal which starts out from here. And this corresponds to an error in the insert size. Right. So no more about this. I'm running out of time anyway. I will skip this. So just as a recapitulation, here is the general structure. And over this talk, I have told you that this general structure actually consists of many different steps. So first you do error correction, then you construct the de Bruijn graph. You try to cope with the errors in the de Bruijn graph. Then you go to repeat resolution which uses read pairs. You estimate distances, you resolve repeats, you close the gaps, then you refine your contigs, and you get the final result. And this picture here shows the new stuff that SPAdes introduced along this process. So there is a Bayes hammer tool for error correction. There are new scheduling policies for correcting the errors here. There is a novel tip clipper that uses gap closing. There is bulge core removal with additional bookkeeping. There are special algorithms for chimeric removal. There is clustering for estimating insert sizes. There are paired De Bruijn graphs as the basic underlying model for repeat resolution. There is a new gap closer here. And there is a parallel project which copes with the rectangle graph. Then there is a new tool, SQL, which was not developed by us, but which was developed in UCSD. And finally, we even have a new quality estimation tool, which introduces better, more accurate metrics for measuring the quality of the assembly, which I will not go into now. All right. So basically, I have tried to convince you that SPAdes has many novel ideas, and now the only thing left to convince you in is that these ideas work. And here are some on the other hand, some empirical results from single-cell data sets. On single-cell data, well, this is one of the data sets. You can see that SPAdes outperforms both conventional assemblers like Velvet and SoapDenova, and these assemblers with some modifications for the single-cell case. So we have bigger NJ50, and what's even more important in this case, we have higher genome coverage, because this is the main problem. Like Velvet has only one misassemble, but it has very low genome coverage. All right. Somewhat surprisingly, SPAdes also beats conventional assemblers in the multi-cell case, because we have made it a bit we have made it more accurate, and it turns out maybe that in the multi-cell case also there are some situations where you have to be accurate. So we see that there is slightly better coverage and larger N50 even in the multi-cell case. And yes, of course. Yes, sure. Here is a sample genome map. You can, I'm not sure you can see anything specific, but what you can see is that there is this coverage gap, which of course nobody can do anything with. But on these low coverage regions, you can see that SPAdes is a bit greener than other tools. Okay. These tables are the table on the left. I think you cannot read this, but this is the number of complete genes captured by SPAdes, captured by different assemblers. And you can see that SPAdes is roughly on par with IDBA, a bit more, but this may be, maybe random. And here is the percentage of the genome mapped, and again, SPAdes kind of beats the competition. This is the NG plot. I do not have time to explain what it means if you don't know already, but higher is better and SPAdes is higher. This is the NGA plot, which is a modification of the NG plot. And I think I have run out of time, so I will be closing down. We have several ongoing collaborations on single-cell sequencing projects with various institutes and various teams around the world that try to apply single-cell sequencing to real-life problems, sequencing pathogens isolated from the hospital environment. A very important project, marine bacteria, marine samples that also contain, which are also a metagenomic sample where you cannot cultivate an individual bacteria, but for some reason, I don't know why, it turns out that they are much more successful in producing antibiotics and in pharmaceutical applications. So there are a couple of projects about sequencing marine bacteria, etc., etc. And we are slowly going into comparative genomics, so we are trying to modify SPAdes to do a comparison between two genomes or a genome and a set of reads and stuff like this. Okay, thank you for your attention. And these are